Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mehboob. In our last class, we were discussing photosynthesis in higher plants. In that, we covered the structure of chloroplast and various chlorophylls present in higher plants and algae as well. Uh, Let us have a look. So, this is the structure of chloroplast, the ultrastructure of the chloroplast. Uh, we learned that the light reaction occurs in grana and the dark reaction of photosynthesis occurs in stroma. So you can see the thylakoids arranged in a group, piled up thylakoids you can see here. So these are the sites for light reactions. Okay, and stroma is the site for dark reaction. What happens in the light reaction? ATP and NADPH, that is the driving force, the assimilatory power is produced during light reaction. Okay, so we also discussed that there are nine types of chlorophylls are present in all green plants. In higher plants, you can see chlorophyll A and chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. You can see in bacteria and lower plants, and bacterial chlorophyll A, bacterial chlorophyll B, chlorobium chlorophyll 650, chlorobium chlorophyll 660. You can find them in bacteria. Okay, so we saw the empirical formula, the difference between the chlorophyll A and chlorophyll B. We said that the chlorophyll has a porphyrin structure with the four pyrrole rings. See, these are the pyrrole rings. Pyrrole rings are nothing but four carbons with the nitrogen, five five atom chain. This is a closed chain. See, here is the nitrogen and four carbons attached to the nitrogen. It's a pentane structure with the nitrogen at its center. Okay, so uh, you have all the four. This is the uh, first pyrrole ring. This is the second pyrrole ring, and this is the third one. This is the fourth one. At second pyrrole ring, in chlorophyll B, instead of CH3, you will find CHO, formyl group. Otherwise, everything is similar to that of chlorophyll A. And this is the terpene alcohol, a phytol tail. Okay. This is how it looks in a molecular model. We also learned about carotenoids. We saw differences between carotenes and xanthophylls. Today, we will see the nature of light. You all know that the radiant energy that is coming from the sun includes the most powerful gamma rays, X rays, UV rays, visible light, infrared rays, microwaves, and radio waves. Okay, infrared, microwaves, and radio waves, they have uh, the wavelength is very much greater, so they do not cause any harm. But gamma rays, X rays, they are very powerful. UV rays too. And in between these UV rays and infrared rays, we will find visible light. We all know that in visible light, we have seven colors VIBGR, that is violet, blue, green, yellow, orange, and red color is seen in the visible light. Okay, so visible light has a wavelength of 400 to 700 nanometer. The energy is present in the form of a packet called quantum. The energy in the sun rays, we call it as photon. Each photon consists of an energy equal to h nu, where E is the energy, h is the Planck's constant, and nu is the frequency. So E is equal to h nu is the equation to find out the energy of a particular frequency ray. Okay, so these when a chlorophyll molecule absorbs light quantum, that is photon, the molecule gets excited, and it is brought from its normal ground state to an excited state of energy. And when the light source is turned off, the excited molecule reverts to the ground state again. While returning to the ground state, the packet of energy observed is lost in the form of light or heat. The light emitted on decay of excited molecules is called fluorescence. Okay, so excitation of molecules by light and the fluorescent decay are extremely fast process. You cannot imagine how fast they can occur. About a few picoseconds, that is 10 to the power of minus 12 of a second, it will take to emit the uh, fluorescent light. Okay, so this is how this packet of energy which is absorbed by the chlorophyll molecule, the chlorophyll molecule gets excited and it transfers the electron to the electron carrier. Okay, so absorption spectrum and action spectrum. What is absorption spectrum and what is action spectrum? Chlorophyll pigments observe strongly violet, blue, orange, and red wavelengths, while carotenoids observe only the blue and violet wavelengths. When pigment extract is dissolved in an organic solvent like ether and light rays are allowed to fall on this solution, some specific wavelengths of light are observed by the extract solution, whereas other wavelengths are transmitted through it. The relative absorbance of various wavelengths in a purified pigment can be measured with the spectrophotometer. The observed rays are graphically represented and is called the absorption spectrum. Okay, that is the absorption spectrum. Chlorophyll A shows maximum absorption at 660 nanometer, and other at 430 nanometer. Chlorophyll B has absorption peak at 640 nanometer and the other at 453 nanometer. And the all accessory pigments such as carotenes and xanthophylls have absorption bands lying in between 430 and 490 nanometers. The relative effectiveness of various wavelengths in producing a specified response is obtained from action spectrum. The action spectrum studies led to the following revelations regarding photosynthesis. What are those things that were revealed by the action spectrum? Photosynthesis consists of two light reactions, one activated by red light and the other by far-red light. In terms of oxygen evolution, red light is the most effective wavelength, and a comparison between the action spectrum and absorption spectrum suggests that chlorophyll A is the most efficient pigment of the photosynthesis, and chlorophyll B or phycobilins take second place in efficiency. So these are the things that were revealed by the action spectrum. And the carotenoids are poor supporters of photosynthesis. Then the action spectrum of photosynthesis, what we can see here, the quantum yield curve shows a dip in the region of 450 and 520 nanometer. And here, maximum quantum yield, that is the number of oxygen molecules released per light quanta observed, occurs in between 600 and 680 nanometer. At wavelength longer than 680 nanometer, there is a sudden drop in quantum yield. This phenomenon is known as red drop, and it was identified by Emerson and Lewis in 1943. We will see what is Emerson's enhancement effect and what it revealed. Okay, now Emerson's enhancement effect. Emerson showed that relatively low photosynthetic efficiency of chlorella in far red light, that is in far red light it has low photosynthetic efficiency, and can be increased by simultaneous application of short wavelength, that is far red light and short wavelength red light, if supplied simultaneously, then the yield is more than the individual yields together put together. Okay, so in other words, if x is the rate of photosynthesis in one wavelength and y is the rate in another wavelength, that is x is the rate of photosynthesis in far red light and y is the rate of a photosynthesis in shorter wavelength, uh, and z is the rate where both the short and far wavelength lights are simultaneously provided to it, then z is greater than the sum of x plus y, and it is called Emerson's enhancement effect. And what does this Emerson's enhancement effect reveals? Um it opened the door in understanding the role played by chlorophyll A, chlorophyll B, and accessory pigments in photosynthesis. It is now confirmed that two separate groups of pigments cooperate in photosynthesis, and these are designated as photosystem 1 and photosystem 2, or pigment system 1, pigment system 2. So this pigment system 1 is activated by light with wavelength longer than 685 nanometer, and pigment system 2 is activated by 680 nanometer less wavelength light. Okay, so we will see what are these pigment systems. Pigment systems or photosystems comprise pigment protein complexes associated with reaction centers. You can see here, this is the pigment system, light harvesting complex we call it. Okay, it has nearly about 240 molecules. So if it is pigment system 1, then the reaction center, it observes the uh, longer wavelength light, that is 685, and if it is the pigment system 2, it is it observes the light at 680 nanometer. Okay, so all these accessory pigments, they absorb the light energy and they transfer these light energy to the reaction center through a resonance transfer, and the reaction center gets excited and it releases a electron, or it loses the electron, uh, which is transferred to the primary acceptor or accepted by this primary acceptor. So this is the light harvesting complex we call. See, this is the energy transferred, it is in the form of photons, and the energy packet, we call it as quantum, and this quantum of energy is absorbed by these molecules and get excited, and the electrons in the outermost orbit, they will be lost, and they are transferred to the primary acceptor. Okay, so the pigment system 1, it contains chlorophyll A. You can see here, pigment system 1 contains chlorophyll A and less chlorophyll B. It has beta carotenes and proteins. It occurs in the unstacked stroma lamella. One of the chlorophyll molecules is a special chlorophyll, that is the reaction center. It absorbs light at 700 nanometer and it is called P700. Okay, it is the reaction center, and all the surrounding chlorophyll A and beta carotenes in the photosystem, they transfer their energy. Uh, then the pigment system 1 contains iron sulfur proteins, which are the primary acceptors of electrons transferred from the reaction center. So the PS1 generates a strong reductant capable of reducing NADP plus and a weak oxidant. So this is the pigment system 1. And the pigment system 2. Pigment system 2 contains a chlorophyll A, more chlorophyll B, beta carotenes, and proteins. It is located between closely stacked grana. See, you can find the difference here. Pigment system 1 is present in the unstacked stroma, whereas pigment system 2 is present in the stacked grana. Okay, then it is and is not in direct contact with stroma. This this stacked lamella is not in direct contact with the stroma. The reaction center is P680 here, and photosystem 2 also contains the primary electron acceptor called pheophytin. Pheophytin is a molecule of chlorophyll without magnesium ion. Okay, it is also a type of chlorophyll molecule, but no magnesium ion is present in pheophytin. Pheophytin and P680 and protein are bound to quinone, that is Q pool. Then PS2 also contains protein bound to manganese called manganese protein. So these are the two pigment systems. One more time, we will check the differences between chlorophyll, this pigment system 1 and pigment system 2. Pigment system 1 contains more chlorophyll A and less chlorophyll B, and beta carotenes and proteins are uh, present. And here in pigment system 2, it contains less chlorophyll A and more chlorophyll B. Beta carotenes and proteins are also seen here. It occurs in unstacked stroma lamella, pigment system 1, and pigment system 2 occurs in stacked lamella, that is in grana. The reaction center in the pigment system 1 is P700, and the reaction center here in pigment system 2 is P680. And here the primary acceptor is the iron sulfur protein, that is ferredoxin, and here the uh, primary acceptor is the pheophytin, which is just like a chlorophyll molecule but without magnesium ion. Okay, so these are two pigment systems. Then photosynthetic unit. A photosynthetic unit is defined as the smallest group of collaborating and cooperating pigment molecules necessary to affect a photochemical act, that is, absorption and migration of a light quantum to a trapping center. About 400 chlorophyll molecules constitute a photosynthetic unit. The tight arrangement of chlorophyll molecules allows for excellent energy transfer by resonance transfer. These tightly packed light harvesting chlorophyll molecules are also called antennae chlorophyll. Now we will see what are quantosomes. And these quantosomes are considered that photosynthetic units exist as morphological, densely pigmented granular structures in the thylakoids. These distinct morphological structures are called quantosomes, the name given by Park and Beggins in 1964. However, the quantosome concept of photosynthesis is not accepted because the thylakoids lacking quantosomes still carry out photochemical production of oxygen. Okay. Then there are different electron carriers in the photophosphorylation system, that is cyclic and non-cyclic photophosphorylations. Before going to that, we will just see what are different electron carriers present in this photophosphorylation systems. Okay, the first one is plastoquinone. Plastoquinone is present in the chloroplast as large pools fixed with the membranes. They are considered as hydrogen carriers in oxidation reduction reactions. Plastoquinone links pigment system 2 to cytochrome B6 and F complex, which in turn interacts with the pigment system 1 through the mobile plastocyanin. Then the cytochromes. Cytochromes are extremely important carriers of electrons in both chloroplasts and mitochondria. They are made up of iron containing proteins. These cytochromes are made up of iron containing proteins. The iron can alternate between the ferric and ferrous states depending on oxidation reduction reactions. The cytochrome B6 F complex is a supramolecular unit embedded in the thylakoid membranes. It is composed of iron sulfur proteins bound to cytochrome F, cytochrome B, and plastoquinol. Then the one more electron carrier you can see is the plastocyanin. Plastocyanin is a copper-containing protein. It appears blue when oxidized and colorless when reduced. It is bound loosely to the inside of the thylakoid membranes, that is the side towards the lumen away from stroma. So iron sulfur proteins, these proteins are considered to be the most primitive of all electron transport mediators since they have low molecular weight. They exist as either membrane bound or in a free soluble state. They accept one electron at a time, and three classes of iron sulfur proteins exist. 8 iron and 8 sulfur, 4 iron and 4 sulfur, 2 iron and 2 sulfur iron sulfur proteins are seen. Okay, out of these 8 iron and 8 sulfur are found in bacteria, whereas 4 iron and 4 sulfur are found in chloroplast, and 2 iron and 2 sulfur in the soluble ferredoxin of chloroplast. Now we will see ferredoxin. Ferredoxin is a protein that contains iron and sulfur. Reduced ferredoxin reduces NADP plus. The enzyme that mediates is known as ferredoxin NADP plus reductase. It is a flavoprotein that uses NADP as an electron acceptor. Okay. Now, uh, we will see redox potentials. Photochemical reaction in photosynthesis has two functions to carry out. They are to drive the electrons from H2O to NADP, that is photolysis of, after photolysis of water, the electrons and protons are carried out, they are transferred to photosystem, electrons are transferred to photosystem 2, and the protons are transferred to NADP to reduce NADP to NADPH, and to provide energy for formation of ATP by establishing the proton gradient. So the oxidizing agent in oxygen evolution is provided by the pigment system 2. The reducing agent required for reduction of NADP plus is provided by pigment system 1. The electron transfer pathway involves both photosystem 1 and photosystem 2. Okay, now we will see photophosphorylation, that is light reaction. This was first proposed by or demonstrated by Arnon in isolated chloroplasts. He demonstrated that isolated chloroplasts produce ATP in the presence of light. They term the process of ATP synthesis in chloroplasts as photosynthetic phosphorylation or photophosphorylation. So it is of two types: the cyclic photophosphorylation and the non-cyclic photophosphorylation. See, a photochemical phase, that is light phase. Light phase has this photophosphorylation, that is cyclic and non-cyclic photophosphorylation, and the dark phase, that is reduction of carbon dioxide to glucose. Okay, so the cyclic photophosphorylation. In cyclic photophosphorylation occurs in chloroplasts illuminated with far red light. Under this condition, only photosystem 1 is activated. You can see here. See, P700 alone is activated. Okay, for red light. Okay, then cyclic phosphorylation is important because it provides a source of ATP that is independent of the production of NADPH. You cannot find here any NADPH formation, but only ATP formation can be seen in cyclic photophosphorylation. When NADPH accumulates and the amount of ATP produced during the flow of electrons from water to NADP plus is inadequate to drive the dark reactions of photosynthesis, this cyclic photophosphorylation provides a source of ATP. That is, if NADPH accumulates more and required ATP are less, then this cyclic photophosphorylation supplies the required amount of ATPs. Okay, so what happens here, we will see. When photosystem 1 is activated by one photon, the excited electron is lifted from its reaction center, that is P700. See, this is the P700, the reaction center. So one of the electron gets excited. The electron passes through a chain of electron carriers with increasing reduction potential. Okay. So the first carrier is designated as A0, which is known as the iron sulfur protein, and then the electron is moved to ferredoxin, from ferredoxin to cytochrome B6, and from cytochrome B6 the electron enters into plastoquinone pool. This is plastoquinone pool. This is the reduced plastoquinone, and this is the oxidized form plastoquinone. Okay, so here um, the protons are released into the lumen of thylakoid membrane. See, you can see here these protons which are present at the uh, which are drawn from the stroma to reduce this plastoquinone. When this plastoquinone undergoes oxidation, these H plus ions are released into the lumen. Okay, so electrons are subsequently transferred to plastocyanin from cytochrome F. Okay, then ultimately they reach back to the P700. Here you can see that uh, in the plastoquinone pool, hmm, where from cytochrome B6, this plastoquinone absorbs the electron. At the same time, it absorbs two protons from the stroma. It gets reduced, and this reduced plastoquinone, when it undergoes oxidation, again it loses the protons, and these protons enter into the lumen, and the electron moves on to plastoquinone through cytochrome F, and ultimately it reaches the P700. Okay, it results in setting up a proton gradient and subsequently formation of ATP takes place. The formation of ATP by the cyclic electron transport is called cyclic photophosphorylation. And during cyclic photophosphorylation, ATP is produced, but no oxygen is released, and NADP is not reduced. You can see here no oxygen, no photolysis of water occurred, and no NADP formation also seen. Only ATP is formed. Now we will see non-cyclic photophosphorylation. And this scheme was proposed by Hill and Bendel. This scheme is known as Z scheme of photosynthetic electron transport. It involves both photosystem 1 and photosystem 2 acting in series connected by electrons transport mediators. Uh, the process begins with the absorption of light energy by light harvesting complex. It is transferred to P700 by resonance transfer. See, you can see here the P700, the reaction center. It absorbs the required light energy through resonance transfer, and one of its electrons gets excited, and this electron reaches the primary acceptor iron sulfur protein. Okay, the electron process through a chain of intermediate electron carriers to a soluble ferredoxin present in the stroma. The first carrier A0 appears to be a chlorophyll A monomer, whereas the second carrier A1 is probably a phylloquinone. Okay, so ultimately the electron from iron sulfur protein reaches the plastoquinone, and plastoquinone from plastoquinone, the electrons are supplied to NADP. At the same time, from stroma, it gains two protons. It absorbs two protons from stroma. So this NADP is reduced to NADPH. What happened here? Initially, one more time, both the pigment systems take part in non-cyclic photophosphorylation, and this scheme was proposed by this Hill and Bendel. And here, uh, initially, P700 gets excited by absorbing the light energy, and the electron is transferred to iron sulfur protein, and this electron moves on to plastoquinone, and later on these electrons are supplied to NADP, which gets reduced by absorbing two protons from the stroma, and NADP is converted to NADPH plus. Okay, so where does the electrons lost by this P700 will be fulfilled or supplied? Okay, so we will see. Now at the same time, the P680 also gets excited, and it also loses one electron, and this electron in the as the case of cyclic photophosphorylation, it moves from pheophyte into plastoquinone pool, and plastoquinone first it was in the oxidized state, and it gets reduced by absorbing two protons from stroma, and converts itself into plastoquinone reduced, and this plastoquinone reduced, it again loses electron to cytochrome B6, and at the same time it loses two protons, and these two protons enter into the lumen, where the concentration of H plus ions increases, which ultimately leads to the formation of ATP, and the electron which is released by this plastoquinone is absorbed by cytochrome B6, and through cytochrome F and plastocyanin they reach P700. So the lost electron from P700 is supplied by P680 through these electron carriers. But here it also lost one electron. So where does this electron it will get? So here the most important photolysis of water takes place. This process you can see only in non-cyclic photophosphorylation, and photolysis of water it occurs in the presence of manganese ions and Cl minus ions. Okay, so water splits into two H plus ions and two electrons and oxygen atom, that is of O2, and this oxygen is evolved into atmosphere, uh, which is released during photosynthesis. Whatever oxygen is released during photosynthesis is due to photolysis of water, and this photolysis of water occurs due to the driving force of it is in reduced form, this P680. So it drags the electrons from water molecule nearby water molecule. This water molecule splits in the presence of sunlight and in presence of manganese and chlorine ions, and it splits into H plus ions and two electrons and a half water molecule. So this water molecule is liberated into atmosphere, and whatever electrons are lost by this P680 again reach the P680. So this is very important process, photolysis of water, which ultimately supplies the electrons as well as releases oxygen into the atmosphere. So this is about today's class. Tomorrow or we will see the next things. Okay, thank you very much.